I had the privilege to present our phase 1 data on a novel antibody drug conjugate targeting the protein claudin-6. It's a novel target. It is the first in human study. We've treated 81 patients in a dose escalation and dose expansion study, and this is the first presentation of this data. Claudin-6 is a tight junction protein that is generally not expressed on normal tissue, but expressed in some cancers. Most importantly, in ovarian cancer, it's associated with adverse outcome, shorter progression-free and overall survival. We've become interested in this protein because it's not expressed in normal tissue. If you look at the publicly available GTEx data that allows you to interpret the expression of receptor or surface proteins in normal tissue, you can see that claudin-6 is absent in normal tissue, as opposed to other targets that we're pursuing with ADCs, such as TROP2 or HER2 or folate receptor, which is expressed in normal tissue. So the hopes are that by targeting claudin-6, we will have a bigger therapeutic window to minimize toxicities and deliver chemotherapy payload with greater precision and efficacy, with less toxicity. The study has enrolled now 50 ovarian cancer patients, and we're pursuing a registration strategy in ovarian cancer with a soon-to-start phase 2 study. The overall safety data look very promising because we've had two main toxicities. One is neutropenia, which occurred within the dose escalation at a dose of 3.0 milligrams per kilogram. We then expanded a cohort with pegfilgrastim and were able to completely ameliorate the hematologic toxicity. The other side effect has been neuropathy, which is a known potential side effect of the payload, which is MMAE, which is a vendotin. The good news is that it only occurred in mild severity, grade 1, grade 2, across the now 81 patients that have been treated. There's only one patient that had grade 3 neurotoxicity, which was a young male patient with a germ cell tumor that had extensive prior treatments with bone marrow transplant and platinum-based therapy. So overall, very well tolerated. Particularly noteworthy is that we didn't see ocular toxicities and we did not see pulmonary toxicity, probably attributable to the fact that claudin-6 is just not expressed in normal lung tissue. The efficacy is promising in the sense that overall, depending on the dose cohort, we saw response rates of patients that had less dose than 2.4 of 30%. Those patients that had being treated with 2.4 or 3.0 had response rates above 40%. If we look at a subset of ovarian cancer patients, likewise, those that were platinum resistant, not more than 3 prior lines of therapy. So the target population, the response rates were either 52% or 42%. Small numbers, albeit very promising data. Also very durable responses over time, as I was able to show with the spider plots. Thus, a clear signal to take this forward into phase 2 study. So in summary, targeting claudin-6 with the ADC TORL-1-23 with a MMAE payload with a drug-antibody ratio of 4 is a promising new approach in a subset of patients with ovarian cancer that have high expression of claudin-6. It has shown a very favorable toxicity profile, and it has shown very promising activity, and we're now launching a global phase 2 study with 80 international sites that will test 2 doses moving forward, 2.4 and 3.0 milligram per kilogram, and which will hopefully lead to a randomized phase 3 study for platinum-resistant recurrent high-grade serous ovarian cancer. I think we've all witnessed, the significance of antibody-drug conjugates that allow us to deliver chemotherapy with greater precision and less toxicity We've recently seen the success of mirvetuximab soravtansine as the first approved drug for platinum-resistant ovarian cancer. However, this drug is only available for about 25% of high-grade serous ovarian cancer patients that express very high folate receptors. So we do need other ADCs that target other populations, and it's noteworthy that the expression of claudin-6 is inversely correlated with the folate receptor, so that patients that have low folate receptor expression generally have very high levels of claudin-6 expression. So we're targeting a slightly different patient population with claudin-6, which I think will add to the armamentarium that we have for platinum-resistant ovarian cancer. Also, I do think we need to focus closely on toxicities that we do see with ADCs, particularly pulmonary toxicities with pneumonitis, ocular toxicities, and therefore I think developing strategies that target a very clean target in the sense that's absent in normal tissue and only expressed in malignant tissues as claudin-6 are important strategies to really develop a new generation of ADCs with less toxicity and drugs that can be given chronically without cumulative toxicity over time.